All right, so let's dive into this topic. Antibiotic resistance. It just seems like it's constantly evolving. You know? It's like we're in this arms race, right? But it's a microscopic one.
Yeah.
Where bacteria are finding these new ways to kind of evade the drugs that we use.
Mm.
And for you guys out there, medical microbiologists, understanding these mechanisms is critical for everything you do.
Absolutely.
So we're going to be pulling some insights from this book, Antibiotics, Challenges, Mechanisms, Opportunities. That's by Walsh and Wensfitch. And, I was reading through it, and I got to say, some of the stuff that these bacteria can do is unbelievable.
It really is. It really is. It's fascinating how they're able to adapt.
Oh, and survive.
Yeah. And the first stop on our deep dive here is going to be bolactam resistance. So these are the antibiotics, that we use all the time. Your penicillins, cephalosporins, carbapenems.
Right.
They all rely on that lactam ring.
Yeah. And it's, that analogy of the spring-loaded warhead for that Milacam ring is pretty accurate.
Well, because the way that ring structure is, it's under tension. It's ready to just pop open and attack those penicillin-binding proteins, the PBPs.
Right. Okay, so walk me through this.
Yeah.
How does that attack actually happen at, like the molecular level?
So basically, it all comes down to a sil transfer. So what happens is the bolactam ring will bind to a very specific amino acid residue in the active site of the PBP. And this is usually a serine.
Okay.
So when it binds, they form what's called a penicilloyl enzyme complex.
So is it kind of like a molecular handshake, but it goes wrong?
Exactly. But the thing is, instead of letting go, that bolactam just holds on so tight.
Oh.
It forms a stable bond, and essentially it jams up the PBP.
And that stops it from being able to build the cell wall.
Exactly.
Yeah.
And without that cell wall, the bacteria can't survive.
Right.
It's a pretty elegant mechanism, I think.
Elegant, yeah, from a scientific standpoint, I guess. But not so elegant when you're a patient dealing with one of these infections, right?
Yeah. No, definitely not.
But it is amazing that, these bacteria have evolved their defenses, right,
Yeah.
against these antibiotics.
Oh, absolutely. So they've developed these enzymes, these lactamases that can break down lactamase.
Really?
So it's like they're one step ahead of us almost.
So how do they even, evolve to have lactamases?
Yeah.
I mean, how does that even happen?
So it's really a classic example of just natural selection. Bacteria are always mutating.
Yeah.
And so in an environment where there are lactams present, the bacteria that have the mutations to produce these lactamases, they have the survival advantage. So they get to pass those genes on, and then over time, that's how you get these highly resistant populations.
So it really highlights the importance of stewardship.
Right.
With antibiotics. The more that we use them, the more we're driving this evolutionary pressure.
Exactly. And it's not just about overuse. It's also about, I'm thinking back to a few years ago. There's a patient who had a really complicated UTI.
Okay.
And it was caused by E. coli. And it was resistant to a lot of things. It was resistant to cephalosporins. And we finally figured out it was producing an extended spectrum balactamase, an ESBL.
Yeah, those ESBLs. Those are becoming a huge problem because now they're breaking down even the newer cephalosporins that we have, right? Like ceftriaxone, ceftazidime.
Exactly. And then on top of that, you have the carbapenemases,
right,
like your KPCs and your NDMs.
Right.
And those are even more worrisome because
absolutely.
They can actually inactivate carbapenems, which are kind of like our last resort.
Yeah, right. It's our last line of defense against a lot of these multi-drug resistant bacteria.
Right.
I remember reading a report actually about a hospital that had an outbreak of KPC producing Klebsiella pneumonia. And it was so hard to contain.
Right.
And unfortunately led to, several deaths.
And that is just one example of how these carbapenemase-producing organisms are emerging more frequently. And it's a huge concern for hospitals everywhere.
So what can we do? I mean, are we just at the mercy of these superbugs?
Not at all. Scientists have been hard at work developing new strategies. One of those strategies is to develop lactamase inhibitors.
So it's like fighting fire with fire, I guess.
In a way, yeah. Clavulanate, sulbactam, tazobactam, those are all examples of these lactamase inhibitors. What they do is they bind to the lactamase, and that prevents it from breaking down the lactamase antibiotic.
So that's how those combination therapies work, like augmentin and zosyn, right?
Exactly. So you combine like amoxicillin with clavulanate or piperacillin with tazobactam, and that way you're protecting the antibiotic. It's not being degraded.
Gotcha.
It can reach its target.
But, bacteria, they're crafty.
Yeah.
So I imagine they're already finding ways to get around these inhibitors.
You're absolutely right. We are seeing increasing resistance even to those combination therapies.
So what's next? What's the next step in this whole microscopic chess match?
Well, researchers are looking at a lot of different things. One of the things that seems to be pretty promising is the development of new balactamase inhibitors, like avibactam. And avibactam is part of this combination drug called Avicaz, and that's shown to be really active against some of those really difficult to treat carbapinimase-producing bacteria.
So it sounds like we're making progress, but it's a race against time, right?
It is, absolutely. But it's not just about developing new drugs.
Right.
We also need to focus on preventing the spread of resistance through things like responsible antibiotic use, and infection control measures.
Absolutely. Okay, let's move on to another really interesting example of resistance. Let's talk about streptogramins. So these are the antibiotics like Sinersid.
Right.
And they target that bacterial ribosome. So that's like the protein-making machinery of the cell, right?
Exactly, yeah. And the way they work is they actually bind to different sites on that ribosome.
Okay.
And essentially, they just throw a wrench in the protein synthesis process.
But doesn't targeting the ribosome affect the bacteria's own protein synthesis?
Yeah.
How do they even survive that?
That is a fantastic question, and it really just highlights how incredibly adaptable bacteria are. So while streptogramins do bind to bacterial ribosomes, they actually have a much higher affinity for those ribosomes compared to our own ribosome.
Oh, okay.
So it's all about selective toxicity.
Right. So it's like a really carefully aimed dart.
Exactly.
You're hitting the bacteria, but you're trying to minimize the damage to the host.
Yeah, minimizing any collateral damage.
Exactly.
But of course, bacteria have figured out ways to resist even that.
I mean, yes. They found a way to modify the ribosomes.
You're on the right track. But it's not just about modifying the ribosomes. They have a couple of tricks. So remember Sinersid, it's actually a combination of two components.
Right.
Dalfopristin and quinopristin.
Right. And each one targets a different spot, right?
Exactly. Different sites on the ribosome. And bacteria have evolved these distinct mechanisms to inactivate each one of those components.
Oh, wow.
So for example, dalfopristin can be inactivated through acetylation.
Okay. So they're attaching an acetyl group to the dalfopristin.
Yeah.
And that stops it from binding?
Prevents it from binding to the ribosome.
Gotcha.
And then for quinopristin, they actually have this lyase enzyme that breaks its cyclic structure.
Oh, wow.
So it's like dismantling the key to the lock.
It's amazing how they can target each piece of that drug so specifically.
Isn't it?
Yeah.
It's really remarkable.
Yeah, these bacteria are resourceful. All right, ready to move on to our final example for this this part of our deep dive.
Absolutely.
Let's talk about aminoglycosides. So these are the powerful antibiotics like gentamicin, dipромycin.
Right.
And again, like streptogramins, they target the bacterial ribosome.
Yep.
But these microbes, I bet they figured out a way to resist these as well.
Oh, you bet they have. And they don't just rely on one strategy. Aminoglycoside resistance is like multifaceted.
Multifaceted, all right.
So they've developed all these different mechanisms to evade these drugs. One of them is increased efflux, where they can actually pump the antibiotic right back out of the cell.
So like they have a little bouncer at the door that's kicking out all the aminoglycosides.
Kicking them right out. And then another thing they can do is actually modify their ribosomes in a way that reduces the aminoglycoside binding. But I think their most impressive trick is enzymatic modification of the aminoglycoside itself.
So now they're not even changing the target. They're changing the weapon.
Exactly. And they've got this whole arsenal of enzymes that can add these different chemical groups onto that aminoglycoside molecule. So you've got things like your acetyl groups, your phosphate groups, and adenyl groups.
And these modifications, that stops them from binding to that ribosome.
Exactly. They disrupt those really delicate interactions that are needed for that antibiotic to latch onto its target.
Oh, wow.
And what's really interesting is that we can actually track a lot of these modifications back to specific genes. And the source actually goes into some detail about this with examples of how kanamycin is modified. And it's amazing how these really tiny, subtle changes can have this huge impact on resistance.
Yeah, like they figured out the code of our antibiotics and they're rewriting the rules of the game.
I like that, yeah. But we're not giving up. Scientists are coming up with new strategies to fight back.
So what's next in the fight against aminoglycoside resistance? Are there like new things in the pipeline?
Yeah. There are some really exciting developments. One of those is plasmomycin. It's this next generation aminoglycoside, and it's actually been designed with specific modifications to evade those common enzymatic inactivation strategies that bacteria use.
So they're like tweaking the structure of the plasmomycin to outsmart the bacteria.
Exactly.
That's fascinating. So it sounds like we're making progress, but it's a constant, back and forth, right?
It's an ongoing battle.
Yeah, and that's what makes this field so challenging and so important.
Absolutely.
We're always learning, adapting, trying to stay, one step ahead of these microorganisms.
That's a fascinating field. So are you guys ready to put those medical microbiology brains to work?
Oh, yeah.
All right, here's your first question. Which class of antibiotic is inactivated by lactamase enzymes? Is it A, aminoglycosides, B, lactams, C, streptogrammins, or D, tetracyclines? I'll give you a minute to think about that.
Yeah, I'm sure our listeners out there are already coming up with their answers. But while they're thinking about that, let's move on to question number 2.
Okay.
All right, this one delves into the mechanism of action for a specific lactamase inhibitor. What is the mechanism by which clavulanate inhibits lactomasesis? Is it A, competitive inhibition, B, allosteric inhibition, C, mechanism-based inactivation, or D, covalent modification of the active site serine? Choose wisely.
That's a tough one.
It is.
But don't worry, we'll go over all the answers in just a bit. All right, for question 3, we're switching gears to streptogrammins. Specifically, which of the following is a mechanism of streptogrammin B, also known as quinupristin resistance? So is it A, acetylation, B, phosphorylation, C, lyase-mediated linearization, or D, methylation of the ribosome?
Keep those brains working, everybody. We're almost there.
Almost there.
Question number 4. This one takes us into the world of aminoglycosides. Now, these antibiotics are often our go-to for serious infections, but as we talked about, bacteria have a lot of ways that they can become resistant to them. So tell me, aminoglycosides are primarily inactivated by enzymes that catalyze which of the following modifications? A, glycosylation, B, hydroxylation, C, decarboxylation, or D, acetylation, phosphorylation, and adenylation?
All right, last one. This one focuses on a next-generation aminoglycoside. So we're talking about plasmomycin. How has plasmomycin been designed to overcome resistance? Is it by A, targeting a different site on the ribosome, B, blocking efflux pumps, C, inhibiting bacterial protein synthesis by a novel mechanism, or D, evading common enzymatic modifications?
All right, everybody. Pencils down. Let's take a second to catch our breath, and then we'll jump into those answer explanations.
Sounds good. Ready to see how you did?
Oh, I'm sure everyone got 100%. But even if you missed a couple, it's all part of the learning process.
Well, exactly. It's all about learning.
So let's go through those answers and see what we can learn. All right, so question 1. Which class of antibiotic is inactivated by lactamase enzymes? The correct answer is B, lactams. And this is because lactamase enzymes, they're specifically designed to target that black tamarine. They break it open, and then the antibiotic is basically useless.
It's a perfect example of how smart bacteria are. All right, question 2. What is the mechanism by which clavulanate inhibits lactamases? This one was a little tricky. The answer is C, mechanism-based inactivation.
I remember how we were talking about how clavulanate, it's like that suicide substrate.
Yep.
It basically tricks the lactamase into binding to it, but then it forms that stable covalent bond.
Right.
And that just shuts the enzyme down for good. It's like a little molecular trap.
It's a really good trap.
It is. It is. Okay, question 3. Which of the following is a mechanism of streptogrammin B or cunipristin resistance? The answer here is C, lyase-mediated linearization.
Yes.
And that's all thanks to that VG-biolase enzyme that we were talking about earlier.
It's a tough one to beat.
It is. It basically just breaks that quinipristin molecule and then it can't bind to its target on the ribosome.
Exactly. All right, question 4. Aminoglycosides are primarily inactivated by enzymes that catalyze which of the following modifications? This one, if you remember those 3 main enzymatic modifications we were talking about, the answer was pretty clear. It's D, acetylation, phosphorylation, and adenylation.
Yeah, they're really good at using these chemical modifications to just completely disarm those aminoglycosides. It's pretty impressive when you think about it, how they've evolved to do that.
It is. It is. Okay, last one, question 5. How has plasmomycin been designed to overcome resistance? The answer is D, evading common enzymatic modifications. So Scientists basically modified the structure of plasmamycin to prevent those enzymes that bacteria use from inactivating it.
It's like they gave plasmamycin a little molecular shield.
Yeah, I like that.
So it can sneak past all those defenses and get to its target on the ribosome.
Exactly. And there you have it. That's the end of our little quiz.
How did everybody do?
Hopefully you learned a few things.
Yeah, and it's a good way to, really solidify those key concepts that we've been going over today. But before we wrap up completely, I want to leave our listeners with something to think about. We've been talking about all these ways that bacteria resist these antibiotics, but I'm kind of curious what all of this means for the future of medicine.
That is the question, isn't it? This rise of antibiotic resistance, it's a huge threat to global health. We're already seeing these infections that used to be really easy to treat, and now they're becoming much harder to manage.
Yeah, it's kind of scary to think about, what if we get to a point where the antibiotics we have just don't work anymore?
Well, and that's what we're trying to avoid, right? Because it could mean we're going back to that pre-antibiotic era.
Oh, wow.
Where simple infections, could be deadly again. And surgeries, transplants, even routine procedures would be so much riskier.
Right, because we rely on these antibiotics for so much.
Yeah.
It's hard to imagine a world without, effective antibiotics.
Yeah.
So what can we do? How do we prevent that from happening?
Well, it's going to take a lot. First of all, we've got to promote responsible antibiotic use.
Okay.
Overprescribing and misuse are huge drivers of resistance. We need to make sure that antibiotics are only prescribed when they're absolutely necessary, and that patients take the full course of treatment.
So it's not just about, coming up with new drugs. It's about how we're using the ones that we already have.
Exactly.
Yeah.
But we do need to invest in research and development for new antibiotics and even alternative therapies. That pipeline for new antibiotics, it has really slowed down in recent years. We've got to reinvigorate that effort.
What about, you mentioned these alternative therapies. What are some of the things that are being looked at?
So there's some really cool stuff happening with phage therapy, where we're using viruses to kill bacteria and antimicrobial peptides. And those are naturally produced by our own bodies. Those are showing some promise, too.
So it sounds like we kind of need to explore every option.
Right. We do. And another thing we can't forget about is infection control measures in healthcare settings.
Right.
Preventing the spread of resistant bacteria. That's just as important as developing those new treatments.
Absolutely.
Yeah.
So it's really like a team effort. We need the researchers, the clinicians, policymakers, even the public.
Absolutely.
All work together on this.
It takes a village.
It does. And that includes our listeners out there, right? You guys are the medical microbiologists. You're on the front lines. And your expertise in identifying these resistance patterns, helping to guide treatment decisions, it's critical.
Yeah, you guys are vital in this fight.
Well, this has been a really interesting deep dive. It really highlights how complex, and how urgent this antibiotic resistance problem is, and how bacteria have all these incredible ways of evading the drugs that we use and the challenges that we face in trying to stay ahead.
It's a constant arms race.
It is.
We have to be really vigilant and proactive to combat resistance.
I hope this deep dive has been helpful to you guys out there. Maybe you've learned some things that you can apply in your work.
Yeah.
And maybe it sparked some, further interest in this area, because it is a really important area of medicine. This is a battle we can't afford to lose. So keep learning, keep asking questions, and keep pushing those boundaries of knowledge. And we'll see you guys next time.